Hello students, welcome in chemistry class. Biomolecules, one of the important unit which is related to our life. So we will discuss with the help of different slides. Biomolecules means the compounds which are present in living organisms. These compounds are mainly made by carbon. There are a number of elements present in the environment, but the versatile character of carbon made it very important element for living organisms. Some of the compounds present in living organism is known as cellular pool. There is a list of different elements present. In biomolecules, there are two types of compounds, inorganic compounds and organic compounds. Easily you can see that inorganic compounds are available less gases, water, but organic compounds have their major importance. Carbohydrates, proteins, fats, enzymes. There are number of biomolecules which are made by carbon and I think made a complete life. There are two types of biomolecules, micromolecules and macromolecules. Micromolecules are always small in size and have lower molecular mass. For example, water, minerals, some gases, etc. While macromolecular biomolecules have large molecules with large size as well as higher molecular mass, like carbohydrates, proteins, vitamins, like that. There are number of biomolecules. Here a list is given. What are the building blocks of those biomolecules and their functions? Like carbohydrates. Carbohydrates are energy giving food material or energy giving biomolecules. Their building blocks are monosaccharides. Protein, body building materials. Amino acids are their major constituent. DNA, deoxyribonucleic acid, which is one of the substance which transfer the heredity character from one generation to another generation. Their monomers are nucleotides, while RNA, which is responsible for protein synthesis, its monomer is also nucleotides. There is a slight difference that we'll see in a coming slides. Carbohydrates, the important biomolecules, energy supplements, and even storage in our body also. We'll see the details of these carbohydrates, which is one of the important biomolecule. Carbohydrate, important biomolecules. Carbohydrates are mainly known as hydrates of carbons. They contain normal formula, general formula CnH2n1. In general, it is told that in carbohydrate, the ratio of carbon, hydrogen and oxygen is in 1 is to 2 is to 1 form. Some of the example we have like C6H12O6, C5H10O5, their ratio of carbon, hydrogen, oxygen is in 1 is to 2 is to 1 form. Carbohydrates have so many important function, not only energy giving, but they are body former also. Body former in the sense with the protein, they form some exoskeletons, cell walls, cellulose is there in the plant which made cell membrane. So carbohydrate is very important for living organism. Now if we want to classify the carbohydrates, on what basis we will classify? If we see the first classification of carbohydrate on the basis of hydrolysis product. On the basis of hydrolysis product, carbohydrates are classified into three major categories. Monosaccharides means which cannot hydrolyze further. Second, oligosaccharides means they have number of monosaccharide units, like disaccharides have 2 monosaccharide units, trisaccharides have 3, and so on. Polysaccharides always have more than 10 monosaccharide units, like starch, cellulose. As we discussed that all the carbohydrates made by monosaccharides. Now, how many types of monosaccharides are available? There are n number of monosaccharides are there. We can classify these monosaccharides also by two ways. On the basis of number of carbon atoms, like triose sugar has 3 carbon, tetrose sugar 4 carbon, pentose sugar 6 carbon, hexose sugar 7 carbon, like that. On the basis of functional group also, these monosaccharides can classified as aldose and ketose. If there is a aldehyde group with polyhydroxy group, then they are called aldose sugar. And if there is ketone with hydroxyl group, then they are called ketose sugar. Oligosaccharides are made by the combination of monosaccharides. On the hydrolysis, they give number of monosaccharide units. Like disaccharides provide 2 monosaccharide units on hydrolysis. Trisaccharides gives 3 monosaccharide units, and tetrasaccharides give 4 monosaccharide units, and so on. Oligosaccharides generally contain 2 to 9 units of monosaccharides carbohydrate. Disaccharides, there are mainly 3 disaccharides are there, like maltose. Maltose made by alpha glucose, 2 units of alpha glucose. Lactose made by glucose and galactose monosaccharide. Same way sucrose, which is one of the most important disaccharide, as a sugar we are using it, it is made by glucose and fructose. Polysaccharides contain more than 10 monosaccharide units. As we know about starch, glucose, these are the important examples of polysaccharides. Polysaccharides are also of two types, homoglycans and heteroglycans. Homoglycans means they contain only one kind of monosaccharide units, like like starch, while heteroglycans containing more than one type of monosaccharide units. One of the important category of the polysaccharide, storage polysaccharides, means food is stored in this form in living organisms, like glycogen in human body. Extra glucose, extra sugar is deposited, storage in form of glycogen. Inulin is one of the storage carbohydrate. Starch, especially in plants, carbohydrates are stored in form of starch. Another category of polysaccharide is structural polysaccharide. For example, cellulose available in plants to make outer layer, to make protective layer, for example, cell wall. Chitin is another important carbohydrate which is used to make exoskeleton in some of the invertebrates. This is one of the important classification. It is also asked in the board also, number of times it is asked, so please keep in mind. Reducing sugar and non-reducing sugar. Sugar which can oxidized by Tollens reagent and Fehling solution They are called reducing sugars. Like most of the monosaccharides, lactose and maltose, they are reducing sugar. Means they can reduce to Tollens reagent and failing solution, or we could say they oxidize themselves by these two oxidizing reagents. While non-reducing carbohydrates, non-reducing sugars can't reduce to these two reagents, Tollens and failing solution, like sucrose and other all polysaccharides are non-reducing sugars. Glucose is one of the important monosaccharide. It is also known as aldohexose since it contains aldehydic functional group with a hydroxyl group and 6 carbons are available. Two types of structures are given of glucose: linear structure and cyclic structure. Mainly naturally occurring glucose is dextrose, means dextrorotatory glucose. Structure is given in that slide. There are number of evidence to prove the linear structure of carbohydrate. Its molecular formula is C6H12O6. When it reacts with hydrogen iodide, it forms normal hexane. It shows that all 6 carbons are bonded in a straight chain. When it reacts with hydrogen cyanide and hydroxylamine, it produces cyanohydrin and oxime respectively. It shows that there is a presence of carbonyl group in this glucose. When glucose is oxidized in presence of mild oxidizing agent, it forms gluconic acid which has 6 carbon. It means the carbonyl group is available in form of aldehydic functional group. When glucose reacts with acetic acid, its acetylation taken place and 5 acetic acid molecules are consumed, pentaacetate glucose is formed. It means there are 5 hydroxyl groups. There is a one alcohol group is in primary form. It means primary alcohol is available which can be proved with help of oxidation reaction in presence of nitric acid. When it is oxidized in presence of nitric acid, glucaric acid or saccharic acid is formed. But open chain structure cannot verified by the following reactions. Like glucose cannot react with Schiff reagent, while other molecules can react. Glucose cannot react with 2,4-DNP, cannot react with sodium bisulfite, while other aldehydes can react. Its a pentaacetate form cannot react with hydroxylamine, and even its linear structure cannot show mutarotation. It means no change in optical activity. It means glucose has some other structure as well. Cyclic structure of glucose can show all these properties which cannot be shown by linear structure. So glucose, it's suggested that glucose has cyclic structure also. In this structure, its a cyclic structure is given in alpha and beta form. Why alpha and beta form? Since glucose molecule has one configuration, either dextrose or levose, but when it comes in a cyclic form, there is a change in configuration at first position only. Maybe at first position, H will be at left side and OH will be at right side, or vice versa. Such kind of isomers are known as anomers, in which configuration is changed only at a first carbon. So glucose shows two anomers, alpha anomers, in which H is at left and OH will be at right. While in beta anomers, H will be in right and OH will be at left. Here in this slide, Haworth formula or Haworth structure of glucose is mentioned, which shows the cyclic structure of glucose in form of pyranose ring structure. 5-membered closed ring structure, that is pyranose ring structure. So energy biomolecule is over. That is carbohydrate which provides us energy. But to get energy, we have a well-built body. And body is built by protein, which is body building biomolecule. So we should see now about the protein. Protein is one of the important biomolecule since it is a functional and structural unit of living beings. Even in plants also, in animals also, especially animal body is made by proteins since we could not have cellulose. These proteins are made by amino acids, means on hydrolysis they give us amino acids or they made by amino acids. Proteins are polymers of amino acids. In our body, 20 amino acids are required. Out of them, 10 are essential amino acids. Essential amino acids means they are not formed in our body, they have to be taken from outside. While 10 amino acids can be prepared in our body with the help of cell materials. These proteins are very important since they are forming our body. Without body, we can't exist. Some are forming different body parts. Some are forming transporting reagents like in our blood, hemoglobin is there. So in one of the other way, proteins are most important biomolecule. Amino acids, as we know by the name now, amino and acid means it contains two functional groups, carboxylic acid group as well as amino group. These amino acids are very important since they combine, they form polymers which is protein. Amino acids show different behavior on the basis of available amino group as well as carboxylic group. Sometimes these amino acids are neutral like glycine if the number of amino acids and carboxylic acids are same in number, or they may acidic or basic. With the help of amino acids only, proteins are formed. Here different amino acids are given. Basically 3 categories are there. Neutral amino acids, if amino group and carboxylic group are same. Acidic amino acids if carboxylic acids are more in compared to amino group, or basic amino acids if NH2 group is more than carboxylic acid group. Aromatic amino acids, alpha amino acids, beta amino acids, like that there are different categories of amino acids with examples are given. In this slide you can see the different structures, and with the help of structures you can clarify how amino acids are acidic, basic or neutral. There are structures of different amino acids. Amino acids form Zwitter ion. Zwitter ion means ion which contain positive and negative both the terminals together. Since amino acids have both the terminal, NH2 group shows basic behavior and COOH, carboxylic acid, shows acidic behavior. And we know that according to Bronsted-Lowry concepts of acid and base, acids are proton donor and bases are proton acceptors. So it may be available in form of NH3 plus ion and COO minus ion, means one amino acid may have both the positive and negative terminal, means they are available in form of Zwitter ion. Now, could you see that if zwitter ions or amino acids which are available in form of zwitter ion applied with electric current, so in which direction they will move? No, they will not move towards any one of the electrode since they are having both the poles in one molecule. So, such pH in which amino acids can't move towards any one of the electrodes in electrical field, that is called isoelectric point. Zwitter ion and isoelectric point, these two are very important points which may asked in exam. So please keep in mind zwitter ion means having both the terminals positive and negative, and isoelectric point means the pH at which mostly amino acids are available in form of zwitter ion and they do not move towards any one of the electrode. Now question comes how amino acids are combined, how they are joined together. Amino acid molecules are joined together with the help of peptide linkage. Part of amino acid NH2 combines with COOH part of another molecule, release water molecule and form NHCO linkage. It is known as peptide linkage. When two amino acids combine and form peptide linkage, they are called dipeptides. And in such a way, number of peptide linkage are formed, they are called polypeptides. There are different structures of protein: primary, secondary, tertiary and quaternary. The most basic structure is a primary structure of protein in which only there is a sequence of proper amino acids which form responsible protein. Primary structure of protein is responsible for functioning of the protein. Another structure of protein is secondary structure. There are different types of secondary structure: alpha helix structure, beta pleated structure and collagen structure. Alpha helix structure, as by the name you can say helix kind of a structure, in which a single strain of the protein shows the hydrogen bonding. Different peptide linkage shows intramolecular hydrogen bonding and coiled. Because of the coiling, they form helix kind of structure. In beta pleated structure, as by the name, they are in a layered form. Different chains of amino acids or different peptide chains, polypeptide chains, are attracted by each other by hydrogen bonding or by disulfide linkage or by van der Waals attraction force. Even dipole attraction force also can work. So different polypeptide layers come closer to each other and they form layers. That is called beta pleated structure. In tertiary structure, the helix is coiled too much, the extent of hydrogen bonding is too much, so that will come in a rod form of structure, and due to hydrogen bonding, helix comes closer to each other. In quaternary structure, number of helix come closer with the help of hydrogen bonding, coiled, too much coiled, and that's why globular form arises there. So tertiary and quaternary proteins or quaternary structures almost equal to the globular protein, or we could say the globular protein has a quaternary structure. In this slide, examples of proteins are there with the different shapes. Now, by the combination of amino acids, proteins are formed. Proteins, there are different types of proteins. How are they in chemical behavior? What are their functions? What are their classification? These many things we have to see in a further slides. This is the functional classification of protein. What are the functions of protein? Some proteins are structural, some are enzymatic proteins, means working as a biocatalyst. Genetics proteins used to transfer the hereditary character. Some are transporting proteins, some are storing protein. So in this slide, number of classes are there of the protein with examples. Please go through it. One of the important classification of protein on the basis of its composition. Some proteins are simple protein. It means they contain only amino acids. While some proteins are conjugated proteins or complex protein, they contain non-proteinous part also with them. Means except amino acids, they have some other parts. Like glycoprotein has carbohydrates, lipoprotein has lipids, hemoglobin has iron. Except amino acids or non-proteinous part of such kind of protein is known as prosthetic group. Some are derived protein, means they derive by the hydrolysis of the protein, like peptones, polypeptides and peptides. They are also known as derived protein because they produce by the hydrolysis of that respective protein. Here one more category is there of protein: fibrous protein and globular protein. Fibrous protein means in which amino acids are there in a linear form. For example, myosin protein in your muscles, fibroin in silk. Globular proteins have tertiary or quaternary structure. They are in a globular form, like fibrin in your fibrinogen, means in your blood. Fibrin is there, which is in a globular form. Hemoglobin contains globin protein, which is in globular form. Egg has albumin protein in globular form. So this one is a major classes of the protein. Conjugated protein may be available in a linear, means in a fibrous form or globular form. Now one of the important thing I think you might have observed, when egg is boiled, it becomes harder. What will happen here? Egg contain albumin protein. When it is heated, the nature of albumin is changed. That type of the process in which the nature of protein is changed due to heat, maybe due to change in pH or due to any chemical, that is known as denaturation of the protein. In denaturation of protein, only biological structures of protein is changed, but no chemical properties are changed. Actually, the stability of the helix or structure of protein depends on its coiling. When temperature or pH change, it's stably affected a lot. Because of the change in pH or temperature, uncoiling taken place, and by this uncoiling, tertiary structures convert into primary structure. Due to change in temperature, pH or in presence of any chemicals, uncoiling taken place. This is taken place due to breaking of hydrogen bonding or maybe of covalent bonding. And because of this only, the biological activities of proteins are affected. That is only known as denaturation. By this denaturation, mostly the structure converted into primary, which is most unstable structure of the protein. Nucleic acids, one of the important biomolecules, they made by DNA or RNA. They are called nucleic acids since they are acidic in nature. DNA is a deoxyribonucleic acid containing different nitrogenous base, pentose sugar and phosphate group. . nitrogenous base, pentose sugar, and phosphate group. Altogether, they form nucleotides, which are the monomers of DNA. All the nucleotides are bonded with each other by phosphodiester linkage, while the nitrogenous base are bonded with each other with the help of hydrogen bond. When nucleotides are linked together with diester linkage, they form single helix. And this single helix combine with the help of hydrogen bond with its complementary thread, and structure will convert it into double helix structure. RNA is another important nucleic acid. It's a long chain nucleic acid, single helix nucleic acid, which is important for protein synthesis. It is kind of messenger which convey the message with the help of DNA which type of protein will form. RNA is same important as DNA. RNA single helix structure. There is also combination of phosphate group, pentose sugar and nitrogenous base. The difference is that only at the place of thymine there will be uracil. Means adenine will be connected with uracil at the place of thymine. And same cytosine will be bonded with guanine. There are different types of RNA. Transfer RNA carries the amino acids. Messenger RNA encodes the combination of amino acids to form protein. Now these amino acids will be transferred or translated to ribosomal RNA. Ribosomal RNA form ribosomes to produce the protein, ribosomal protein. One more type of RNA is there, that is small nuclear RNA, which make complex with protein, especially in eukaryotic cells. In this figure, one nucleotide unit is presented. There is a linkage between phosphate group, sugar, and nitrogenous base. In sugar, always the phosphate linkage will be formed at 5 and 3 carbon. Third 5 carbon, always the combination will be at the same carbon of pentose sugar. And in this slide, different pentose sugar and nitrogenous base are mentioned. As we know that two types of sugars are there, deoxyribose sugar in DNA and ribose sugar in RNA. Their diagrams are there. And nitrogenous base. Two types of nitrogenous base we have, purine and pyrimidine. Altogether 5 nitrogenous base we have, adenine, thymine, guanine, cytosine, and for RNA, uracil. There are different nucleotides by the combination of phosphate group, nitrogenous base and sugar. Different nucleotides are formed by combination of different types of nitrogenous base. Since phosphate group and pentose sugar will be same, there will be difference only in nitrogenous base. There is the complete structure of DNA, double helix structure. Watson Crick proposed this structure. In this double helix structure, helix are formed by phosphate group and ribose sugar. But the ladder of this double helix are formed by nitrogenous base. As I told that in nitrogenous base, combination will be always same, like adenine will be combined with thymine and cytosine will be combined with guanine. You can see in this way, the number of adenine always will be equal to the thymine and cytosine will be equal to the guanine, always in any DNA. But yes, the number of AT and CG may differ. This combination may differ. And this combination only decides the different genes, different codons, which behaves means decides the behavior or the nature of the proteins, and which will decides different DNA fingerprints. Different RNAs are given, messenger RNA, transfer RNA, and ribosomal RNA. These structures are mentioned here. But for both purpose, these are not important, but we must know, since RNA is very important for protein synthesis, it's very true that without DNA, RNA also cannot form protein synthesis. DNA has a special characteristic to replicate. Means if during division, if one helix is removed, DNA can form its complementary helix with the help of nuclear material. DNA is available in nucleus of the cell. RNA is already single helixed, available in cytoplasm, and there, with the help of combination of amino acids, it forms protein. I hope this video will help you to prepare for biomolecules. Almost the things are in short, but key points are included. Go through the video, make your notes according to that, and please like and subscribe the channel.